going on. And what appears to be going on is that hydrogen sulfide, unlike cyanide, one of the things that it does is it very rapidly depletes a particular tripeptide. I'm going to show you the structure. It's a fairly simple molecule. Let me rotate this a little bit so you can see it. This is a tripeptide. It's 3 amino acids that are strung together. On this end over here, we have the amino acid glutamic acid right here. It's a carboxylic acid, CO2H, CH, NH2, CH2, CH2, and then there would be another carboxylic acid. That's coupled to another amino acid, which is called cysteine. And here you can see that. That's got an SH group, a free thiol or SH group here. And then that is also coupled through this carbonyl. We call these things peptide bonds. These CO and H groups. You'll see there's one here, and there's another one over here. This is a tripeptide. There are 3 amino acids, 1, 2, and 3, that are strung together through these 2 dipeptide bonds. And the last amino acid here is glycine. So this particular thing is called glutathione. And glutathione is a very, very important biological molecule. It does a lot of different things. And when you deplete it, it's no longer there. One of the functions of glutathione is to protect you against things like free radicals. And free radicals are highly reactive species that, for instance, they're implicated in aging processes. In some cases, they might be implicated in cancer. There's a whole bunch of things that free radicals can do, but they really wreak havoc with the body. And one of the things that this particular material, this glutathione, does is it actually neutralizes or eliminates these free radicals. So when it's depleted, it's not able to do that. Another function is that it detoxifies organic and inorganic xenobiotics. So these are both organic molecules and inorganic molecules that are not actually normally present in the biological makeup of an organism, but that are introduced into its environment. And for instance, things heavy metals, which would be what we would call inorganic xenobiotics, things like lead and mercury and arsenic. These kinds of materials will actually bind lead and arsenic and mercury will bind to this sulfhydryl group here, to this SH group. So in that way, they're actually removed and rendered, they can actually be removed in their soluble forms and be neutralized and be rendered non-toxic. It's important to have glutathione. It's also essential for the function of the immune system. It induces lymphocyte proliferation, and it's also involved in apoptosis, which is the process whereby cells are actually programmed to die at a particular rate. We have programmed cell death. I think I mentioned this once before. Cells, or at least for those of you that took this last year, you know about apoptosis because there is actually a mechanism whereby cells are not supposed to proliferate indefinitely. They, after a certain number of divisions, they're actually programmed to die. So that actually is interfering with that. And the other thing about glutathione, it's involved in DNA synthesis and also the repair of DNA. It's involved in protein synthesis, in the synthesis of prostaglandins, and also in in the activation of a number of enzymes. So it's believed that unlike things like hydrogen cyanide, which just are basically just eliminating or interfering with the activity of cytochrome C oxidase, hydrogen sulfide, on the other hand, not only does it do that, but it also depletes very rapidly glutathione levels, and that makes it particularly dangerous. So why do I tell you all this? Because in order for these organisms to be able to survive under these conditions, in fact, they have to be able to eliminate the hydrogen sulfide. But the hydrogen sulfide, nevertheless, is important because it's actually a source of sulfur, and it's also a source of a particular kind of it's actually a source of energy because it can be used, the oxidation of the hydrogen sulfide, the energy that's produced from that, can be used to make ATP. It can be used for a lot of other processes that would be taking place within the organism, and organisms that are in this particular zone where no sunlight ever falls, and it's impossible for them to be, they are autotrophic, but it's impossible for them to be photoautotrophic because there's no light. They have to develop another system. They have to become chemoautotrophic, so they have to become autotrophs that are deriving their energy from some other means rather than through say the breakdown of water molecules using sunlight. So they're actually using, instead of using water, they're using hydrogen sulfide. But again, there are a number of organisms that are living in these fumarole or vent communities where basically without some kind of protection, these organisms would die. So we've got two things going on. We've got the hydrogen sulfide, which we say is very, very toxic. And the other thing that I was interested in is, since we know that some of these fumaroles can actually heat up to as much as 400 degrees, what kind of temperatures are we talking about in these vent communities? What kind of temperatures do some of these creatures have to be able to withstand? So I want to talk a little bit about both of those things. And I also want to talk a little bit more about the what's actually going on in the trophosome, in this tissue that I mentioned before, this dark green, spongy, lobular tissue that actually constitutes the feeding body of these giant tube worms. So what is actually taking place in there? Well, he said that there's bacteria, and your book actually says that, tells you that there are bacteria, but the book gives you the impression, when you look at it, that the bacteria seem to be more or less free-living, and they're in this space within the trophosome, and they're in all these individual bacteria. And in fact, that's not entirely correct. So that's what I want to talk a little bit about, how these bacteria and how a little bit more about the structure of trophosomes, which was actually elucidated by very, very careful electron microscopy. So by examining these things in great detail, what scientists have found out is that the bacteria actually they're not really free-living, as is shown in these pictures, but they actually exist inside cells that are called bacteriocytes. And these cells are specifically designed, the bacteriocytes are actually produced by the tube worm, but the bacteria live within the bacteriocytes. So they're not, as the picture seems to indicate, just free-living, but there's a bacterium in each one of these bacteriocytes. So, and if we actually look at the structure of these things, we'll find out even the bacteriocytes are more internal and towards the actual side of the trophosome. That is the part of the end of the trophosome that is right up against the ventral and dorsal blood vessels. We have other structures there that are actually called trophochrome cells. And the trophochrome cells, unlike the trophosome, trophochrome cells actually have pigmented bodies in them. And the pigment that's inside these cells is actually giving rise to the green color. So the organism itself, the tube itself seems to be bright red, the plume is red, but the trophosome, the feeding body itself, is actually a rather intense green color because of the pigment that's inside the trophochrome cells. So we have the trophochrome cells on the outside. We have the chrome cells on over here, and then inside, more internal to that, we have another set of cells, which are the cells that are actually containing the bacteria. That is the bacteriocytes. And what it's found is that the bacteriocytes that are in the center of the trophosome are the bacteria that are actually contained in there are smaller and much more biologically active than the older ones. So there's actually seems to be a gradation as you go from the center to the outside. You get less biological activity this way, and more in the center, where the cells are younger and they seem to be much more metabolically active. And we know this because we can look inside the cells and we can see that the cells, as they utilize the hydrogen sulfide and they're actually oxidizing this material, they're actually forming elemental sulfur. And you can actually see particles of sulfur suspended inside the trophosome and inside the bacteriocytes. You can actually see these particles of sulfur using electron photomicrography. So that's kind of an interesting observation. So that's the structure of these things, and again, what's actually happening in there is that the bacteria are oxidizing the hydrogen sulfide. So we can take H2S, and there is actually, it's not really clear what the source of oxygen. It could be molecular oxygen. It could be oxygen from some other species. There is a fair amount of dissolved oxygen, even at that great depth. But I'm just going to show it as, because I don't really know that it's molecular oxygen, but I'm just going to show it as oxygen, and then we would form H2O plus S. So if I really wanted to balance that appropriately, since sulfur is actually in the form of SH, S8, we would probably write 8 H2S plus, and if I wanted to use molecular oxygen, I guess then I would have to write 4 O2, and that would give me 8 H2O plus S8. You can barely see this. Let me try to see if I can write it a little bit larger. 8 H2O plus S8. So that would be a balanced equation that would represent that. There are other sources of sulfur, other forms of sulfur that can be utilized, but this is principally what's coming out of the fumarole, and that's what these organisms are doing. These reactions are also catalyzed by other forms of sulfur. The sulfite is another form. That's so this is sulfide, S, H2S. Sulfide is S2 minus. We also have sulfite. I'm going to erase the glutathione here. We also have, this is sulfide, SULFIDE. We also have sulfite, which is SO3, 2 minus. This is sulfite, SULFITE. And we also have S2O3, 2 minus. This is called thiosulfate. And another form of sulfur is actually sulfate, which is SO4, 2 minus. This is sulfate. These are some of the commonly occurring oxidation states of sulfur. And so there have been a lot of studies to see what, adding thiosulfate or sulfite or sulfide, what that actually does to the rate of carbon dioxide uptake, carbon dioxide fixation.